Good evening, everyone. Thank you so much, Lynn and Dusty and Montessa, for having me on this call. So today I'm going to speak about a lot of what I spoke about at the summit, the LiveLung summit, which I thoroughly loved. I think it was the most gratifying day for me to be with all of you in person. So with that, let me share my screen. Well, as Montessa very kindly introduced me, I'm an associate professor at the Icahn School of Medicine at Mount Sinai. And my lab really focuses on understanding the biology of lung cancer, primarily small cell lung cancer. We want to study what's happening in the tumor. We want to do what's happening in the immune microenvironment. And we use a lot of tools to study that, like a lot of high throughput sequencing, mouse modeling, a lot of molecular biology techniques to study lung cancer, with really the goal of precision medicine, which is not one size fits all. It's giving you the medicine that truly works for you by having a knowledge about your tumor, having a knowledge about the biology of your tumor. I also wear another hat. I'm the director for the Lung Cancer PDX program, which is a patient-derived xenograft mouse model program. For that, we try to collect biopsies and blood samples from patients who are either coming for treatment or are enrolled on clinical trials. And we try to make mouse models from there. Today, I'm going to talk about the translational pipeline that we follow in the lab, because that's really what we do. But not many of you know, the fight for lung cancer is actually personal for me. I've lost a couple of family members to lung cancer, and specifically small cell lung cancer. So this fight is actually very personal for me, and it's kind of my life's purpose. So hopefully today I'll be able to walk you through what we do in the lab and sort of demystify some of what we do. So we follow what we call a translational pipeline. From when a patient comes into the clinic, I'll walk you through what happens when someone comes to you with a consent form to take your tissue or to take your blood. What happens to that tissue or to that blood? And what do researchers like myself try to achieve with the samples that we collect from you? So the first stage is, of course, the diagnosis, which usually happens with scanning, with immunohistochemistry, and then the doctor comes to you with a diagnosis of your disease. So when we think about small cell lung cancer, like when a patient is diagnosed with small cell lung cancer, most patients who come into the clinic are already at an extensive stage disease. Like about 60 to 70% of the patients, about 2/3 of patients already have an extensive stage disease, which means that the cancer has spread beyond the primary space and metastasized locally or to extend to another tissues of distant origin. So they are all treated with a standard frontline therapy with chemotherapy in combination with immunotherapy. Initially, the patient has an objective response rate of more than 60%, but unfortunately, the disease often relapses. And when they relapse, there are options for second-line therapy, but they're quite limited compared to probably other malignancies. The first-line therapy, as I mentioned, is with platinum doublet. So it's etoposide with either carboplatin or cisplatin. And they get two PD-L1 drugs that are currently approved, atezolizumab or durvalumab. And then in the second-line regimen, depending upon whether you've had the chemotherapy-free interval of less than 6 months or more than 6 months, there are several options. Lurbinectedin is approved. Topotecan or irinotecan is approved. There may be clinical trials that are in your space. If you're eligible for that, then you will be enrolled. You'll be asked to enroll in a clinical trial that applies for you. But there are options in the second-line space. So this is sort of the trajectory of immunotherapy trials and approvals. The ones that are in green are the phase 3 trials, and the ones that are in red are sort of this large phase 3 trials that were positive and which led to the approval of the two PD-L1 drugs. So Empower 133 led to the approval of atezolizumab, and Caspian study led to the approval of durvalumab, which is another immunotherapy agent. And these are some of the other trials which also looked at this combination. They're either in Asia or in other countries, and the Keynote 604 looked into pembrolizumab. So this has sort of been the trajectory. The introduction of immunotherapy is quite recent. It has happened in the last 6 to 7 years. Like 2018 is when the data for Empower 133 came out. So it is not it hasn't been there for a very long time, but it has really led to improvements. So this is where we were in 2009, where the only option we had was with the platinum doublet. And the 3-year survival was less than 5%. And essentially, we had no long-term survivors. But to 2022 and 2024 now, I mean, we are seeing the 3-year survival has gone up from 5.8% to 17.6%. And we do see some long-term survival patients. So the 3-year survival is there, and the curves do flatten out. So immunotherapy has a role in a subset of patients, and they do work for this minority of patients. But the problem is, this is truly a minority of patients. So this is sort of the first-line therapy that we have available. So now, when you get a diagnosis and your samples are collected, so there it could be blood, it could be tissue samples. So these tissues are collected from the patients, and then they are taken to the lab to either make cell lines, mouse models, or they're directly taken for doing sequencing studies that I'll talk about in a minute. But usually that's what your samples are collected for, the blood or the tissues, that's what they're collected for. So what we run, and this is done in several centers, is we run a centralized lung cancer PDX program, and we support the thoracic oncology services and to generate what we call a lung cancer PDX platform, which is accessible to investigators of the scientific community. So these samples can be collected from the patients. It could be the surgical, pleural fusion, biopsy, autopsy samples, neural mets, or with CTC, which is circulating tumor cells from the blood. And it is injected into a mouse model, and then we observe tumor growth in the mouse. And why this is important? Because we can do several things from these mouse tumors. So either we can do drug response studies or what we call co-clinical trials in the lab to look for novel therapies that can be applicable for patients in the clinic. Or we can run them through several pipelines like histology, whole exome sequencing, proteomics, RNA sequencing on single-cell RNA-seq. If you read literature, if you read sort of papers in the field, you'll be very familiar with these terms, and I'll walk you through today about what these mean. But these tissues can be used for doing these high-throughput analysis. This is what we do as researchers, is what we call preclinical research. And it is a whole lot of things under this umbrella. So it can be a drug screening in cell lines. It could be high throughput sequencing. It could be what we call a whole genome sequencing, gene editing studies, histology, and then testing in cell lines and in mouse models. And there is a huge component where we integrate the data with computational knowledge that we have, either from publicly available data sets or from AI. And we try to make predictions about drugs, about biology, and what we think could be the biomarkers of drugs in this space. So what have we learned? So this was back in 2020, that we published this sort of histology atlas for small cell with 174 cases, where we stained the tissues that we got from patients with these markers, the AACL1, NeuroD1, PAU2F3, YAP1, which are the subtype markers, or with neuroendocrine markers. Some of these markers you can see in your pathology report. And that's how the diagnosis for small cell is made. What we sort of understood from this analysis is that small cell is extremely heterogeneous, which means it can be driven by more than one markers, or we can get more than one markers in one tissue. And as you can see here, there's these two markers, AACL1 and NeuroG1, in two areas of the same tissue, or we can get tissues which are driven by these specific marker expression, like truly AACL1 driven, NeuroG1 driven, or PAU2F3 driven. So this gave us an idea that small cell is truly small blue cells under a microscope, and they express neuroendocrine markers like INSM1, chromogranin, or synaptophysin, some of which you will see in your pathology report. And it expresses several markers like ACL1, NeuroG1, or PAU2F3, which are sort of the subtype markers. And we understood, which is very important, that small cell is very heterogeneous as a tumor. Now, when we do NGS or whole exome or whole genome sequencing, and you will see, you must be familiar with these terms because the tumors undergo this testing. What we understood is that small cells, so this is sort of what we understand from the mutational profile, is small cell has a lot of mutations, and a lot of these mutations, most of these mutations are loss of function mutations, which means the functions of these genes are lost. And the majority of cases, we see loss of function mutations in these two genes, P53 and RB1. So what we understood from the lessons from the mutational analysis is that small cell lung cancer has very high tumor mutation burden. There are major genetic alterations in P53 and RB1, and we lack targetable recurrent genomic alterations. Like if you think about non-small cell lung cancer, they have these targetable alterations like KRAS, EGFR, ALK, but small cell, unlike non-small cell lung cancer, it lacks these targetable alterations that we can design drugs for. So it's a very different tumor type than non-small cell lung cancer. Now, what we understand from proteomics, it's quite a profound finding. This was published back in 2012 by a team at MD Anderson, and this is the paper. So what they kind of proposed is that small cell lung cancer cell lines, they look proteomically very different from non-small cell lung cancer cell lines, and they have higher expression of several targets. I mean, this is quite a bit of information, but these targets will become later on as I talk about therapies. But they have higher expression of several targets. You may be familiar with the names PARP, ATR, CHEK1. These are all DNA damage repair genes. So the genes that control the repair of DNA in the cells. Now, the lessons that we learned from small cell lung cancer proteomic analysis is that the protein profile is very different from non-small cell lung cancer, and it has a very high expression of proteins that are involved in DNA damage response, like PARP, ATR, V1, and CHEK1. They are major targets that will become major targets for small cell lung cancer. And RNA sequencing really told us a lot. It kind of gave us sort of what we call the genetic atlas or the transcriptomic atlas for small cell lung cancer. And for the first time, we were able to sort of divide small cell lung cancer into these large subgroups that are driven by these transcription factors or the factors that control the expression of genes like ACL1, NeuroG1, PAU2F3, and YAP1. And we also learned that several of these surface markers, DLL3 will become very important because I'm going to talk about DLL3 targeted agents in a few minutes. But we saw that several targets are actually overexpressed in these certain subtypes of small cell. So the lessons that we learned from these RNA sequencing is that the expression of small cell subtype markers like ACL1, NeuroG1, PAU2F3, and YAP1, and expression of the several surface markers or several specific markers, they may or may not be surface markers, like DLL3, immune genes, Notch, and MYC, and expression of markers that could be potentially drug targets, like PARP, EZH2, LSD1, and DNA damage response targets. The newest technology is single-cell RNA sequencing, which means that we are trying to look at the gene expression at the single-cell level. And why do we need the information at single-cell level? So if you think cells as a fruit bowl, when you do this, transcriptomic or bulk analysis, what you're usually making is like a smoothie where it merges the flavor and the color and the taste of all fruits together, and you're not able to look at each fruit individually. But that gives you sort of a gross snapshot of what each cell contribution or the transcriptome of each cell contributions are. So now we go back, when we do single cell sequencing, we are actually able to look at the flavor or the characteristic of each cell, and we are not losing this knowledge or the signaling by making bulk sequencing into sort of what we call a smoothie. So we published the first single cell atlas of small cell lung cancer, and we established that small cell can be divided again into these three subtypes, which is, in our single cell atlas, we saw ACL1, NeuroD1, and PAU2F3. But I think the more profound thing that we understood from this atlas is the contribution of immune genes, how the tumor cells are talking to the immune cells, and what subset of immune cells may be important in having this immunosuppressive tendency for small cell lung cancer. So heterogeneity of small cells that we understood from single cell RNA sequencing, novel small cell subtype, rare cell population. I talked about that smoothie where you lose that, the fruits that may be 1 or 2 in number, you lose that flavor. So we were able to see this rare cell population in the single cell atlas, and we were able to characterize the immune microenvironment, which may be dysfunctional in this tissue. And finally, from the mouse model, and this was a study that was published by an MD Anderson group in 2020, why these mouse models are important is when they tracked the tumors in mouse, they saw that the patient developed brain metastasis, and they could track the same brain metastasis in the mouse model that was made from that same patient. And when they tracked the tumor, how the tumor was responding to the drug, when a tissue was collected from a patient that was responding to chemotherapy, when the mouse model was treated with chemotherapy, they also showed response. So a mouse model can be a very good model to recapitulate the biology of these tumors that we get from patients, and also to do drug response studies that recapitulates how these drugs are going to behave in the clinical setting. So I think that's why it is very important to build these cancer-relevant mouse models, and that's what the tissue is also used for. This is what we do in the preclinical research. And then, of course, the goal is to take this knowledge into the clinic, or how we apply is to how we design clinical trials. So 2 things, we want to have targets and we want to have biomarkers. And you can put your questions in the chat if you don't understand any part of what I spoke today. I'll be happy to answer at the end of my talk. But a target is what you can design a drug against, like PARP and CHEK1. So you're designing drug against. And biomarkers are to select patient population where this drug would show the maximum benefit. So the question is, how do we raise the tail of the curve? Or in other words, how do we make more patients respond to a therapy? So this is sort of the pro and con for immunotherapy for small cell. And I think, there is a research ongoing from a lot of labs, including ours, is to make immunotherapy better in small cell lung cancer. So the pros for small cell is that it has a very high mutational burden. There is genomic instability. So you think that immunotherapy will do better in this disease. But the con is that a lot of these tumors, most of these tumors, have very low or absent tumor infiltrating lymphocytes and low MHC class 1 expression. So this shows, there is an immunosuppressive, or in other words, the tumor cells are sort of hidden from the immune cells. So what I will show you 2 strategies on how we are kind of making immunotherapy better. So I talked about low MHC class 1 expressions. It's, in other words, how to make these tumor cells more visible to the immune cells. So we understand from preclinical studies that these cancer cells are hidden from the immune cells because they suppress this molecule called MHC class 1, which makes the tumor cells more visible to the immune cells. And we published this study back in 2022, where we showed that targeting epigenetic modifiers or epigenetic drugs, which is epigenetic means these targets regulate the gene expression. So these drugs like LSD1 can reactivate or re-express this molecule MHC class 1. There are multiple trials now with LSD1 inhibitor in combination with immunotherapy in small cell lung cancer. The second strategy is that these tumor cells are immune cold, which means they don't have a lot of infiltration of immune cells. So how to make these tumor cells more immunogenic or more visible to the immune system? So for that, we targeted a class of proteins called the DNA damage response inhibitors. So if you think about the cell cycle, I will not go in too much into the detail, but we are targeting this class of proteins which regulates a certain phenomenon in the cell cycle, or we call the cell cycle checkpoint. And these proteins are CHEK1, ATR, and V1. And when you target these proteins, which are either responsible for the repair of your DNA, which is the PARP inhibition, or the proteins that control the G1 checkpoint, G2M checkpoint, like CHEK1, V1, and ATR, you get tumor cell killing. And when you combine these drugs, because they target your DNA, when we combine these drugs with immunotherapy, we see a better response to immunotherapy because it is sort of a synergistic effect. So we published several studies which looks into these DNA damage response inhibitors, which led to several clinical trials. As you can see, the phase 1 trial with talazoparib, which is a PARP inhibitor. And then there are several trials that are looking into PARP inhibitor, CHEK inhibitor, or ATR inhibitor. And then there are several trials that are looking into the combination of these DNA damage response inhibitors with immunotherapy. And I think they're doing well. So there are several of these DNA damage response inhibitors and immunotherapy trials. About other targets, so of course, the efficacy of tarlatamab, which is a DLL3 targeted bispecific T-cell engager. This is the data that was shown in this study, as you can see here. It's a New England Journal of Medicine paper. So it showed a very good response rate, a very promising response rate in patients. So this is a phase 2 trial after 2 or more lines of therapy. And this trial showed an objective response rate of 40% and overall survival of 14.3 months. And the phase 3, which is the Delphi 304, which is a second-line tarlatamab versus the standard of care chemo after frontline platinum-based chemotherapy. This is this trial. You can note this trial number and you can check. And now there are several trials that are looking at this in the frontline regimen. So in the, this is the Delphi 305, which is tarlatamab versus durvalumab after durva plus etoposide in frontline therapy for small cell, extensive stage small cell. And then there is a phase 3 Delphi 306, which is tarlatamab after chemoradiation in limited stage small cell lung cancer. And this is sort of another study, which was also presented at World Lung. This is an ADC or antibody drug conjugate called the ifinatamab deruxtecan, which is a B7H3 targeted ADC in refractory small cell, which also showed a promising response. I'm showing you data from this phase 1-2 study after 2 or more lines of therapy, and the objective response rate was very promising, which is 52.4%. And then there are other studies like the IDA2 and the IDA1, which are looking into this regimen in other setting. And these are the clinical trial numbers. You can, of course, go and check the details of these trials. So this is where we are with sort of the new and interesting. There are several other trials, but I'm just talking about, for the interest of time, about some of the interesting trials in this space. So what about biomarkers? Because we talk about that quite a bit. In non-small cell space, we have several biomarkers like EGFR, like KRAS, like ALK, which helps us sort of devise therapies for these subset of patients. But for small cell, for the longest time, we did not have any actionable biomarkers that could tell us which patient would respond better to which therapy. So in terms of biomarker, we are still definitely not in the same scale as non-small cell, but we are making progress. So let's talk about subtypes of small cell. When you pick up literature, you will hear about these subtypes, which are mainly the ACL1 driven, the PAU2F3 driven, NeuroD1 driven, or the YAP1 driven subtypes of small cell. And they have several accompanying biomarkers or several targets that are higher in these subsets. As you can see, some of the names are mentioned here, like you have DLL3 high in the ACL1 subset, you have NFib or c-Myc high in the NeuroD1 subset, you have ACL2 or SOX9 or MYC upregulated in the PAU2F3 subset, and you have immune genes that are higher in the YAP or the inflamed subset of small cell. And there are other targets also which could be high. So it could be like BCL2 or DLL3, ZH1 and 2 in ACL1, aurora kinase or DNA damage response in NeuroD1. There are several reports that talks about PARP and nucleoside analogs for PAU2F3. And then we have, immune checkpoint blockade or PLK or mTOR inhibitors that have shown benefit in the YAP or the inflamed subset in the preclinical setting. And we also have these tumors that go from the immune cold to the immune hot tumors. So, we are trying to understand the biology at more granularity. And this is one of the biomarkers that has gone all the way from the lab and now being applied in the clinic. So this is a gene called SLFN11. Preclinically, we and others published back in 2016, 2017, that SLFN11 actually predicts response to chemotherapy or PARP inhibitor. And later on, in a retrospective study, it also showed that SLFN can predict response to veliparib, which is a PARP inhibitor in combination with temozolomide. So we were seeing that patients who had high SLFN11 were actually doing better with a combination of veliparib with temozolomide. And based on that, some of these preclinical and clinical data, there was a prospective trial that was designed to look at, SLFN11 as a biomarker for, these patients who received the frontline therapy were then randomized into receiving atezolizumab or atezolizumab with talazoparib, which is a PARP inhibitor, and they were SLFN positive patients. And this study was presented by Nagla Abdelkarim last year at ASCO, and where they showed that the maintenance atezolizumab and talazoparib improve progression-free survival in SLFN positive patients. It is a very modest benefit. As you can see here, the curves, they are not separating out quite a bit, and this was only the progression-free survival. But this showed the feasibility of doing biomarker-driven trials in small cell lung cancer, which I think was a big step forward. And based on this data, there are now several trials being designed or being conceived that are biomarker-driven trials for small cell lung cancer. So what about unanswered questions? There are many, many unanswered questions in the small cell field. So, there's something called lineage plasticity or subtype switching in small cell. So, we are thinking in the preclinical space and also in the clinical space is how the heterogeneity or the complexity of the disease is going to decide therapy. And, how these subtypes change at diagnosis, at progression, and in a longitudinal fashion. And what is the best method to do neuroendocrine subtyping in the clinic? And can subtypes of small cell be a surrogate marker in designing therapies for small cell? And what happens, we talked about SLFN11, what happens in the SLFN low subset in small cell lung cancer? And I think several of these questions, or most of these questions, can be answered by designing prospective trials that are required to compare and confirm the validity of small cell subtypes and methodologies in the clinic. So it's an evolving space, of course, where we go from a completely black box to now we have some biomarkers in small cell, and we are now designing trials based on these biomarkers in small cell lung cancer. And what I believe today I've spoken about is there a cohort of small cell patients that are small but real that have a durable benefit from immunotherapy. And I think there are strategies now to improve response to immunotherapy and define durable benefit. And repression of MHC class 1 is a contributor to immunotherapy failure. So again, designing therapies that can either reactivate MHC class 1 expression or go in a completely MHC independent manner, which is where the BiTEs and the other surface targets have shown benefit, and also DNA damage response inhibitors that can be targeted in that setting that can recruit CD8 positive T cells, and restoration of antigen presentation that can be achieved, as I have shown, at least preclinically, with epigenetic modifiers like EZH2 and LSD1. And of course, personalized biomarker-driven trials are a critical need to determine if the molecular subtypes of small cell are important, and also novel therapies are important, and blood-based biomarkers are becoming very, very important in this space. And the biology of treatment-resistant small cell, I think this is one of the more understudied areas in small cell, is when the patient becomes resistant to the frontline therapy, what's the biology of those patients, and what do we do in the second line and beyond? So the road ahead is improving funding for small cell. So I really, really thank these advocacy groups because they really bring the dialogue to the table of improving funding for small cell lung cancer research. And I think research is truly the way to go forward if we have to have any durable, effective drug targets in this space. I think research has to be like the true north for us. And limited state small cell, I think early detection, interception, and identification of individuals at risk. And I'll talk about the Adriatic trial, which is sort of a very exciting space. New concepts in the works, and developing and implementing effective screening programs. I know small cell lung cancer screening has not been that effective because of how aggressive the disease is, but to continue to have that conversation about developing and implementing strategies to have these patients benefit from screening. And then liquid biopsy and blood-based biomarkers to identify individuals that are high risk of developing small cell. And racial and ethnic disparity in small cell clinical trials, how to make that better, how to identify and disseminate the knowledge gaps that exist that can be implemented to have more ethnic and racial diversity in our clinical trials, and to improve access and enrollment of racial and ethnic diverse populations for clinical trials. And then the potential for bispecific antibodies and antibody drug conjugates, identification of biomarkers and markers of response to toxicity from these BiTEs and ADCs, and then refine these toxicity regimens and adverse events by managing or even coming up with combination therapies. And of course, preventing brain metastases to improve the quality of life, because it is definitely not enough to come up with effective targets which hamper the quality of life for patients. It's not just survival, it's also living, and living in a respectable fashion, having that quality of life. I think it's so, so important. So then having these effective targets that are also improving the quality of life for patients. There are 227 more than 227 trials now that are currently open or pending activation in clinicaltrials.gov for small cell. There are more than 180 currently recruiting trials. This is definitely not a very comprehensive number, so you have to keep updating yourself because there's so much happening. New trials have a trend of this targeted therapies that are independent of pathway activation, so these are more surface targets. And then there are several very, very promising targets in this space, and I'll talk about tarlatamab in a minute, which are targeting these surface targets like DLL3 and SEZ6. And I think we are entering a very exciting space of small cell lung cancer research and treatment. And we are at what we call a golden age. And 2024 alone has shown us that. This is one of the, I think, a month ago, more than a month ago now, this was, this was when I was at that meeting, when this press release came out of improved, this is a limited stage small cell, which looked into durvalumab, either by itself with placebo or in combination with anti-CTLA-4, and they compared it. And this press release showed, and the data is being released at ASCO as a plenary, but this press release showed that durvalumab or Imfinzi, it significantly improved the overall survival and progression-free survival for patients with limited stage small cell. And this is taken from the press release that this is the first and the only immunotherapy to demonstrate survival benefit in this setting in a global phase 3 trials. And it's a positive high-level data that shows a statistically significant and clinically meaningful improvement in the dual primary endpoints of both overall survival and progression-free survival in patients with limited stage small cell who had not progressed following concurrent chemoradiation compared to placebo after the concurrent chemoradiation. So again, this whole data will be released in ASCO this year in a few weeks. But this is an exciting trial in the limited stage setting. And then I think a couple of days ago, we had this release where tarlatamab is now being approved. So FDA granted approval to tarlatamab for extensive stage small cell based on the data that I talked about, based on the trial. And this is sort of the trial design for the Delphi trial that I spoke about. And I think this is a very exciting space, and there are several other interesting trials that are being presented at several meetings. So I think we will see a lot of movement in this space in terms of surface targeting for small cell lung cancer. So I'll end with a message that I always end with is, of course, thanking my team. This is definitely what I gave you today is a highlight of the research from all the investigators. So this is definitely not my research alone, but, all my team members, my collaborators at Mount Sinai, the collaborators all over the world, of course, the patients and their families, none of this would have been possible because all this is anchored in the tissue and the blood that we get from patients. And of course, the funding agencies and the patient advocacy groups. So again, I'll end with a message of hope, because once you choose hope, and I think hope is very, very powerful, but once you choose hope, anything is possible. And I think this would be a good place for me to stop and then address any questions that you may have.
I do have a question for some of the new treatment options and the studies coming out. And we know typically before with the old treatment that small cell lung cancer, of course, had a tendency to come back. So do these, and you showed how it kills the cell, but is it also targeting whatever mechanism caused the metastasis?
So a lot of these targets, we have to see more data on brain mets, but some of these targets do also show benefit in patients with brain metastasis. We will have to review the data, but there are now new drugs coming out that are more brain penetrant capacity, because we understand that brain metastasis is a huge problem. So there are now drugs that are currently coming out that have with more brain penetrance capacity that are currently being developed, but we will have to see how they do in the clinic.
I did post this in the chat, but with the FDA approval of the tarlatamab, and I think it's got a different name. Dusty, thank you for posting that, and Lynn. How soon should we expect to see that being offered in clinic for second line extended stage?
It is now approved, so it should be applied to several. It should be there because now FDA approved, so it should be available. But a lot of these drugs, like the BiTEs and the ADCs, they're pretty involved in terms of the clinical pipeline of patients being treated. So it also depends on the center, and so this will take some time to be implemented, but it's approved. So which means patients will get this treatment in the second line if they are eligible for this trial.
And
For this drug, yeah.
So not everybody, even though it's approved, Dr. Sen, everybody is not eligible for it. Like, say, if I were to go in a clinic tomorrow, have developed, Mets or whatever, I may not be eligible for that, even though it's approved. And what would make me not eligible, or what would make me eligible?
First of all, you should definitely talk to your physician for that. I mean, it is approved for extensive stage small cell, but it depends upon, the patient health. And as I said, this is quite an involved treatment. So it, of course, I'm sure, you'll have to see whether, the health of the patient, and, there are no biomarkers currently, because I don't think patients are being selected based on DLL3 expression, but I think that could be sort of applied. But there are no biomarkers. I think it showed benefit beyond, having a DLL3 marker or not. So I think the eligibility mostly depends upon the health of the patient, the prior treatments that they have received. But it's an approved regimen, so it should be available.
I guess I'm just totally naive on all of this, right? Like, I was diagnosed in 2020, and all I've ever had is etoposide and cisplatin. And, it was pretty rough, and nobody said, oh, my oncologist was like, just, dose her up, she can handle it. So it wasn't like eligibility or anything. So I just was curious on how any of that worked.
I mean, the trial that I showed you, the Delphi 301, it included patients with extensive stage small cell who has received 2 or more prior lines of therapy, which include platinum-based chemotherapy. They are treated, they're stable brain mets allowed. So I think that included like stable brain mets. So, those were, but again, it's a published study. So I definitely encourage you all to kind of read the literature and look at the eligibility criteria. But the best place is to, talk to your oncologist. If there's a relapsed small cell, you should definitely talk about these newer drugs or clinical trials that you may be eligible for.
I just had my second round of tarlatamab, and I hate to use this word, but I feel great. I have been through the ringer. I've seen all the different sauces and spices and sugars and spices that we can throw at one human body. And I'm optimistic, guys. And I just wanted to say that I believe in where this thing is taken us. And I believe that all the research, all of the small cell stuff that a guy like me would never understand. I thank God I have this beautiful red-headed woman next to me because she is my fireball.
What I did find out is, like, obviously it's FDA approved, but that doesn't mean every hospital has it. As of 3 weeks ago, when I went down this to get him on it right away, there are 5 in the country. I have that list. I think Lynn, different people on here know how to get a hold of me. I can give anybody that list, but it has to be a hospital that has already gotten that expanded access through expanded access protocol for this, and that's how we were able to get in right away.
Hey, and also just FYI, if you are interested in information about how to get access to this new drug, please email me or send me a private message, and I can connect you. So just know that
everybody is happy. So I think I answered, but Dr. Sinal, we'll turn the question over to you. Could tarlatamab be used in the first line at some point? And I know it's in the clinical trial.
There is a clinical trial ongoing, and that would be a true game changer if it can be included. And if it shows efficacy in frontline, and it can be included. But there are trials, we don't have the data yet. But of course, fingers crossed. Because the best response that you get up front, of course, it's a no-brainer that the longer the benefit is, the longer the survival, it's always better to get the response from your first line of therapy.
Do you know if Deltra when it might be available in the UK?
I don't. I really don't. I can ask around, and because it just came out, the news just came out. but I can definitely do some digging and then email Lynn or you, Montessa, and then you guys can share with the team.
I have a quick question for Dr. Sen. Dr. Sen, you mentioned the Schlafen 11, and is it similar to like the PDL1 with immunotherapy? No, it's totally different.
No, it's a different. Okay. It's like a DNA RNA helicase. It's a quite complex but it only shows benefit for, not only shows, let me take my word back, not only, but it has, so far the data is, it shows better response. High Schlafen shows better response to chemo or to PARP inhibitor. That's where we have the most data now.
But isn't that similar though with like, if you have a high PDL1, you're probably going to have a better response to immunotherapy, but not so much.
Right. So PDL1, it's like, again, a surface target. It's an immune-based biomarker. Schlafen is not surface, right? So it's something in your cells that then expresses and it predicts response. I mean, in terms of biomarker, yes, it is. If you talk about whether this is like a predictive biomarker, both are predictive. But in small cell, PDL1 is almost always low. So we don't, patients don't get immunotherapy or PDL1 targeted therapy based on their PDL1 status because PDL1 is universally low in small cell.
So I think that's, Wendy, was that a question or they said they might stain the biopsy for DLL3 and that would need to be positive. So is that an absolute for the tartalumab?
No, the data that we have so far, it has worked irrespective of target expression, right? So may not, that is not, of course, I think they will do a DLL3 sort of staining and then look at the expression. But so far, the data that is out there, it shows benefit even with lower DLL3 expression.
And that could have just been to get into the clinical trial. I'm currently under a different trial that's targeting the CES6 expression, and they do not stain for that. That's just 65% of all of us with small cell will have the CES6.
Correct.
But they went ahead on my last biopsies that they took, stained for DLL3, just in case there was a slot that I could go into the tarlatamab trial. But my oncologist, he's the primary at the university, and this is amazing too. I've had 2 infusions and I feel wonderful, you guys, just like Lawrence is discussing with the tarlatamab and Kimberly Gunther, how she feels on that. It's hope, you guys, it's hope, and that's what we're all here for. And I'll find out next week how it's doing, but I feel good, and I know that we're going to have some positive results.
Oh, I have a question at the bottom. I'm sorry. Go ahead, Lawrence or Miss Lawrence.
I just have a question about is the biopsy or the tissue or the blood for the mice, is that something are you in need of the people with small cell lung cancer to send that to you or ask our doctor, or is that something you guys do on your own?
It's usually, center within the center that you're being treated at. If they have a protocol to collect blood or if they have an infrastructure, because it's not just collecting, it's what do you do with it, right? Because these are precious samples. So if they have an infrastructure or someone like me who does research in this area, then they will come to you for consent. They will collect this tissue, this blood. They will tell you that we need this for research. Do you consent to give your tissue or blood? So you would know upfront what your tissue is being used for. Someone will come to you with a consent form and they will say that, hey, we do research and your sample really helps. So would you be willing to give your blood or your tissue for research purposes? So they will tell you, they'll walk you through that and then they will take your sample. But it's usually if they have someone like me who is in the lab who actually does something with this tissue. Otherwise, it's no use, right? For research, they need someone who actually works with them. They will take your tissue for biopsy for clinical diagnosis anyway. So that's a part of the clinical practice. But if they need to take it for research, you will have to give your consent.
Well, we want to thank you so much for coming.
It's a real privilege to be here. Thank you. And Lawrence, I'm really, really happy. I hope that you, Wendy, you all continue get the benefit.